Hello, everyone, and welcome to the Children's Cardiomyopathy Foundation's webinar, Therapeutic Approach to Pediatric Cardiomyopathy Using Gene Therapy, presented by Dr. Barry Byrne. My name is Kristen Dubé, and I am the Family Support and Medical Outreach Director of the Children's Cardiomyopathy Foundation. The Children's Cardiomyopathy Foundation is a nonprofit organization dedicated to finding causes and cures for pediatric cardiomyopathy through the support of research, education, awareness, and advocacy. Established in 2002, CCF has grown into a global community of families, physicians, and scientists focused on improving diagnosis, treatment, and quality of life for children with cardiomyopathy. Your questions are encouraged and welcome during the presentation. Questions can be submitted via the questions box located in the control panel. We will be reserving the last 15 to 20 minutes of the presentation for questions. You are welcome to submit your questions throughout the presentation as you think of them, and we'll go through them at the conclusion of Dr. Byrne's presentation. We will be recording this webinar so that we may offer this to those who are unable to attend live. We are thrilled to be joined today by Dr. Barry Byrne. Dr. Byrne is the Associate Chair of Pediatrics and Director of the Powell Gene Therapy Center at the University of Florida. As a clinician scientist, he studies a variety of rare diseases with interest in developing therapies for inherited muscle disease. His focus is on conditions that lead to skeletal, muscle weakness, and problems in heart and respiratory function. His research group has made significant contributions to the development of therapies for muscle function in patients with inherited myopathies and Pompe disease, a type of muscular dystrophy caused by abnormal glycogen in the muscle. Dr. Byrne also serves as an advisor to the Pfizer Rare Disease Therapeutic Area Advisory Committee and is a co-founder of Avanti Bio, a preclinical stage biopharmaceutical company. Dr. Byrne, why don't you go ahead and get started?
Thanks so much. And I'm glad to be here and share some of what I think is an exciting opportunity for the cardiomyopathy community. I'll just share my slides now and begin this presentation with a look at what the genomics era has brought us. This Time magazine cover really helped establish the notion that we can predict the future by understanding the genetic basis of disease. And although when this was published in 2012, the treatments were not really well solidified about what opportunities there were for actionable treatments based on a genetic diagnosis of a condition in the newborn period. And we know that none of the more severe illnesses that are genetically defined that affect heart function are identified in early life. And this is really ideally at the point which we'd like to intervene. So when you think about the global impact of this in all rare diseases, which are predominantly affecting children, account for about 400 million patients. Those are responsible for a disproportionate number of deaths early in life, and 30% of those children with rare diseases really have early mortality, never reach their fifth birthday, and a large portion of these patients have Duchenne muscular dystrophy, and that's one of the examples I'm going to present as one of the more prevalent causes of early onset cardiomyopathy in children that does also affect skeletal muscle function. And even though, medicine is traditionally focused on small molecules for ameliorating and treating specific disease pathways, when the specific cause of a condition is known, then the genetic material that may be missing or even creating a dominant condition is the best target for treating the root cause of the condition. So even though this type of medication doesn't exist, it is what we would aspire to have, a therapy that is very targeted to a specific disease. So the capsule that we now use, the delivery truck for genetic information, is called an AAV vector. So I thought I'd at least define that for you all, so that that's familiar in this community, because in some other causes of genetic disease, this is now well known. For example, the treatment for spinal muscular atrophy was authorized for commercial use 3 years ago, and even longer than that, there's been clinical research in this area that's focused on addressing the specific genetic cause of type 1 SMA that is identified by newborn screening. So AAV stands for adeno-associated virus. These are small particles that are part of the parvovirus family. And this is a video of what a parvovirus looks like from the outside. And then on the inside, one can remove all the functional genes of the virus and replace them with a therapeutic gene and a promoter that drives that expression. And the only parts remaining are these AAV packaging signals. Those are used to incorporate the genetic material inside this viral capsid, which in the case of early onset or even adult onset cardiomyopathy, the target of cardiomyocytes can be reached through the bloodstream. And so when that virus travels to the to cardiomyocytes through the circulation, it reaches the outside of the cell, traffics through the cytoplasm of the cell, and ultimately leaves its genetic material in the nucleus, where all the other instructions are kept for making proteins in the body. The characteristics of AAV are unique for this purpose, and one of our faculty members at the University of Florida helped understand the structure of AAV, and some of this structural biology work has recently led to a really important discovery for targeting AAV vectors to the heart in particular. By identifying this three-dimensional structure, researchers have been able to add specific ligands that improve the targeting of AAV given in the circulation to go directly to the heart and increase its effectiveness. These are non-pathogenic viruses that are carried along with other viruses like herpes virus and adenovirus in their natural infection state, but those are eliminated in the use of them as therapeutic agents. And there are a variety of types of AAV that were found in nature. And as I just mentioned, there is now the ability to engineer the virus to be even more specific for a given tissue bed. And this has been done very effectively for the heart. When the virus DNA is within a cell, it persists long-term without integrating into the DNA, as some other genetic therapies that are given to cells outside the body, or so-called ex vivo gene therapy, are done. And these long-term persistence is particularly relevant in the heart, where after after children are born, there is really no continued division of the cardiomyocytes within the heart. So those genomes that are delivered or correct these DNA within the cell are effectively permanent, and we'll touch on this in a minute. And of course, there are very few medical therapies that were completely without risk. One of the unique risks of this therapy is it is in effect permanent. This may impact eligibility of participants in gene therapy studies for other studies. And currently it's anticipated for at least central nervous system conditions, and maybe even for heart conditions, that only one treatment might be needed for the lifetime. So this means that one has to think very carefully about participating in a research study to make sure that one's eligible for the commercial therapy. We first discovered the ability of AAV to reach skeletal and cardiac muscle quite a number of years ago now, in 1996, and it's been really taken this long to effectively commercialize these therapies and make new drugs. This is the example of expression of a marker protein within muscle and some therapeutic proteins that we tested over the years in their ability to be delivered by AAV, there is a limitation to the size of a protein that can be incorporated in AAV, and I'll give you an example of that in the work done for Duchenne muscular dystrophy, where the protein is extremely large, the second largest in the body. So at our institution, AAV vectors were really first isolated in the 1970s. It wasn't until 1983 that one of the faculty developed the idea that these could be delivered as vectors. There's a lot more known about their life cycle in the early 90s. And then there was not a tremendous interest until really the early 2000s, when work was being done to show that these vectors could be delivered permanently to skeletal muscle, that there were ability to track the cells that were transduced through a unique combination of AAV and a jellyfish protein called GFP. And then we started to, at our center, develop clinical trials, one for alpha-1 antitrypsin. The LCA trial for a form of congenital blindness was initiated in 2007, and this resulted in the first commercial approval of an AAV vector for inherited blindness called LCA2, or Leber's congenital amaurosis. And the drug is called Luxturna. So then we're close to authorization of a study for a form of severe Parkinsonism. And then a lot of work between 2017 and now is focused on muscular dystrophy. And I'll speak more about that in a second. And ultimately, we hope these therapies actually change clinical practice. This target map shows the ecosystem of AAV gene therapy programs and companies that have helped advance the field with early phase studies, the most outer portion, and then ones that have reached marketing authorization in the center. And from when this map was first created, there's been two new authorizations, and then actually even a third lentiviral vector was approved last week for thalassemia. So as these programs continue to kind of converge on the center with the goal of reaching larger numbers of patients through FDA approval, there are many opportunities here for those indications that affect heart function. Here's just a picture of what these two approved drugs, onosmogene parvovec is Zolgensma for treatment of SMA type 1. This is the first systemically administered gene therapy and the only approved systemic gene therapy product using AAV vectors. The Vitorigine is the Luxturna product I mentioned, just for use in the eye. But you can see the number of interactions with the FDA from this really clinical renaissance of AAV from 2015 to now. Dramatic increases in the number of submissions to the agency. So many studies underway, and several of those new ones are for heart failure. This gives you an overview of the really large population that both adult and pediatric heart failure represented in the EU and the US. 15 million people, and as you see, the really genetic causes constitute a significant portion of that. There are still, I think, more to learn on the etiology of some forms of cardiomyopathy as we get better at screening individuals affected with heart failure and their family members through next generation sequencing. There'll be more potential treatment strategies developed for specific forms of inherited cardiomyopathy. This gives you an idea of the list of genes that are often screened for when a patient presents with either malignant arrhythmia or cardiomyopathy. Much of our work has been focused on acid maltase deficiency or Pompe disease. That's one of the genetic causes of hypertrophic cardiomyopathy listed over here. Some of those other forms of hypertrophy are dominant diseases, and until recently, there weren't easy strategies to manage dominant diseases, but most of the focus had been on replacing a missing gene, like in the recessive causes of Pompe disease. But we now know that there's the ability to both edit and eliminate RNA, specific RNA types through small interfering RNAs that can be delivered by a gene therapy vector. And these would also pertain to some of the dominant causes of severe arrhythmias that in themselves sometimes lead to transplantation, like TMEM43 is one of intense interest right now. So many known causes, as several companies now have identified these and related conditions to target for gene therapy. And in some cases, they're prevalent conditions like in Duchenne. And in some cases, they're really ultra orphan indications where it becomes much more challenging to interest pharma companies in those treatments. So I'll start with the more common one, Duchenne, and talk about another program that's very, very close to our own hearts because of the long association with the patient organization that supported this patient community with Barth syndrome. So I'll tell you about a little bit about Barth syndrome first. So the structural proteins can't be transferred from cell to cell. So every cell needs to get a copy of the gene therapy vector when it's delivered in the circulation, like I showed at the beginning. And dystrophin is a very large protein. It is the second largest gene in the body, but still the largest protein that we express from the genome. And it forms a shock, it has a function as a molecular shock absorber. It links the actin cytoskeleton to the cell membrane through these interactions with membrane proteins called the dystrophin-associated glycoprotein complex. And when those are missing, when this shock absorber is missing, the membrane is subject to tear and muscle injury. This affects early on the skeletal muscles so that boys with Duchenne ultimately lose their ability to walk by late school age 10. And then later in life, they develop progressive cardiomyopathy, and ultimate cause of death in them is due to the cardiomyopathy and respiratory failure. So with a collaboration that we established with a group in Texas, we began to study a microdystrophin construct that removed some of the repeated regions of dystrophin down to a functional version that was identified in patients with Becker muscular dystrophy. This functional version was in many different iterations tested by Jeff Chamberlain at the University of Seattle, Washington. And in collaboration with Joe Cornegay, we began to study the structural integrity of the cells in the golden retriever muscular dystrophy animals that Joe studied most of his career with this construct that was ultimately developed by Solid Biosciences, where I'm the PI of the study conducted by Solid. And here are some examples of 3 dosing groups in the canine model of Duchenne. And you can see that in the vehicle-treated animal, there is no dystrophin or microdystrophin present, but that at the high dose, almost all of the muscle cells could be shown to express dystrophin. And we can see co-localize that with the other proteins of the complex, like sarcoglycan. And when they're merged together, you can see this yellow shows that everywhere dystrophin was present, you can stain for the combination of dystrophin and sarcoglycan so that it had the functional consequence. This is the quantification of the protein, and you can see that it's possible to reach about 2/3 of the wild type level of dystrophin with a single treatment of these dogs at a few months of age. That was an important safety demonstration and efficacy finding that allowed the study to move into humans. And now we have conducted a study with 2 other sites that have enrolled now 9 patients who have Duchenne muscular dystrophy who were treated systemically with this vector expressing the form of microdystrophin called microdys5. And so here's some biopsy information from these patients now, and you can see at 18 months in this first patient who received the high dose therapy, you can see very extensive staining of microdystrophin, co-localization of beta-sarcoglycan, and the presence of nitric oxide synthase, which is believed to be one of the key contributors to improving heart function in this condition. That absence of the ability for the vasculature to respond to exercise is believed to be the cause of functional ischemia, both in the heart and in the skeletal muscles, that contributes to the worsening and loss of function in patients with Duchenne. And you can see that at 18 months, we get rather high percent of positive fibers in patients like this one. And this is the average of patient 4 through 6 in the high dose group. And that this is restoring, again, 2/3 of the wild type level by a Western blot procedure, which quantifies the amount of protein. So very encouraging that we can really just restore all of dystrophin function, keeping in mind this is not the full length dystrophin. So there'll be a need to continue to protect heart function with the medications that are now commonly used in Duchenne. But you can see the consequences in function of the natural history control group declines in function and loses about 100 meters in the 6-minute walk test. And boys with Duchenne around this age may walk only about half the distance of those that are unaffected. And so this gain of 100 meters is very substantial. It would really enable them to function in the community, play sports, do things that kids want to do. One of the subjects in this study is really fond of baseball. He's been able to join a Little League team, something he never would have done otherwise. The North Star Ambulatory Assessment is another set of time function tests that is predictive of the loss of ambulation in Duchenne. And you can see that a significant increase of 4 points on the North Star, 2 to 3 points is considered clinically meaningful. So this represents an improvement in function. And one of the things that we hopefully will demonstrate in this study, because of the long-term follow-up, is that this is durable. So just to summarize what happened in this solid biosciences study, there is stable motor function by 2 measures, the North Star and the 6-minute walk test. All patients showed a improvement in their respiratory capacity as well, improvement in their peak flow and vital capacity. And then patients also report outcomes using a pediatric measure of well-being, the PODCI, and show that they actually have improved activities of daily living and positive outcome from participating in the study. And we will continue to follow these patients for several years to determine the durability and hopefully enroll others using a modified version of what was studied thus far. The other program is for a very small community of patients with Barth syndrome. This predominantly affects the heart, but does have some impact in the skeletal muscle as well. The missing gene is found alike in Duchenne, only in boys. Is called tafazin. And tafazin is responsible for making a critical part of the mitochondria that makes up the mitochondrial membrane. The inner mitochondrial membrane is made up of a lipid called cardiolipin. And without tafazin, cardiolipin is not made, and there's severe mitochondrial dysfunction. This represents probably one of the most severe mitochondrial dysfunction syndromes. And our goal over several years now, both to phenotype the patient population for their cardiac function, which is usually identified in infancy with some degree of heart failure, and then growth retardation, ultimately that catches up later in life. But then in the adolescent years, those boys develop significant worsening of the heart function and arrhythmias that is often leads to transplant. And so we performed a number of preclinical studies that optimized the effectiveness of this therapy to restore tafazin and restore mitochondrial function. So the first paper we published on this topic with Christy Pasek, who's led this work when she was at Florida and a former student, now is at in her own lab at University of Minnesota. We tested a similar concept, a similar vector to the Duchenne one, except the tafazin is small enough that we can fit it in what's called a self-complementary vector that works a little faster. It's a faster onset of action that we can even within a month see the benefits of. And so there were functional benefits, both the mouse heart and the skeletal muscle from this design, testing 3 different promoter elements that control either the native tafazin promoter, one that's specific to heart muscle, or one that's more generic, the CMD promoter. And we found the desmin promoter really provided the optimal expression and biodistribution within the heart. And that's what's been chosen to move forward into the clinic. And with the help of the Foundation for the NIH and the FDA, as well as the NIH NCATS, there is now a new initiative called the Bespoke Gene Therapy Consortium. And our proposed use of this AAV tafazin vector has been nominated to go on to the next stage of selection to be supported by the NIH because of the difficulty in funding the millions of dollars that are needed for each type of a condition that are studied. And with the limited number of patients, that makes it hard to convince a pharmaceutical company to take an interest in this condition. So additionally, we studied how the vector impacts the proteomic activity within the cell. So this is an important part of understanding that the target is effectively engaged, and we've restored tafazin function so that all of the proteins related to mitochondrial function are normalized after treatment. And lastly, we looked at how this works within patient fibroblasts, and we could make fibroblasts representative of both muscle cells or cardiomyocytes, and then study the improvement in mitochondrial DNA because the number of mitochondria now would be increased. And this was a finding in patient-derived fibroblasts with these same constructs that we're testing expressing tafazin. That led us to come up with a clinical candidate, which we've presented to the FDA for consideration in initial meeting, and then through the Bespoke Gene Therapy Initiative, we will move this into the clinic, hopefully within the next 6 to 12 months. An important point to make here is that boys with Barth syndrome have a form of immunocompromise by having diminished white blood cell counts that are periodically rise and fall. And we think, though, that may be helpful, in fact, in preventing rejection of the protein tafazin or of the vector itself. And we'll combine that knowledge about their immune responsiveness with a novel immune management strategy that will prevent reactions to the capsid, which has been observed in some other studies of cardiomyopathy, like the program of Rocket Pharma experienced problems in both adolescent and adult patients with immune reaction to the capsid. And we think we have a strategy now to address that to improve safety. So what's biggest concern about the immunology is that there can be antibodies against the virus, just like we would respond to any viral infection or an immunization for that matter. So how do antibodies influence the success of gene therapy? Well, we know that immune responses can be either immediate or so-called innate immunity. And this is one of the aspects where we think there have been some safety concerns that can be addressed through blocking the humoral immunity, which comes in the form of antibodies against the virus. So in the case of COVID, we all want anti-COVID antibodies, but we don't want antibodies against a therapeutic like AAV. And since AAV is present in the environment, we know that some individuals will be exposed to AAV, particularly in the school age years. And that at the current time excludes those, at least with SMA, where there's a commercial therapy, excludes them from access to the commercial therapy. So in order to make sure this approach is available to as many patients as possible, we've been working hard to understand how to influence the humoral immunity, prevent this downstream activity of that called complement, that is driven mostly by the antibodies that come from adaptive immunity. Cellular immunity can also be an issue, but usually when there is the absence of a gene that's being expressed, that becomes more of an issue to develop chronic inflammation like happens in transplant. And that is more easily managed with some oral drugs. So the strategies to prevent antibodies are a little more complicated, but I'll touch on it. So antibodies are blood proteins that help eliminate foreign proteins. And in this case, the antigen might be the AAV vector. And we know that AAV vectors, if you've never been exposed to them before, will elicit an immune response very soon after they're delivered. And the very first type of antibody to be observed is IgM, which is the one that's most active in activating the complement reaction. So blocking all of these primary responses is a key part of the strategy that we'll use in both in the Barth program and ultimately we think as younger children are treated with Duchenne, this process will be important to sustain the effect and to provide for an opportunity for maintaining the level of expression over the lifespan. If you see one of those antigens, again, you have a lesser IgM response and a much more potent IgG response. And we hope you can avoid this circumstance altogether by treating patients early or preventing their primary response in the first place. We measure those antibodies through a variety of tests that you may hear about for eligibility for a study like in Duchenne or another form of cardiomyopathy. We can test the ability of those antibodies to block the vector from reaching a cell. So these are cell-based assays. They interfere with the binding of this AAV to the cell itself. And these can be influenced by a number of factors in the blood. So these may reflect the ability for the vectors to work in the body, or they may need to be combined with information that comes from this total antibody binding assay called an enzyme-linked immunosorbent assay, or ELISA. The ELISA assay is a little easier to perform. It doesn't require live cells. The participant shares a blood sample. We even think this can be done as a point of care test, like we now test for COVID, and determine whether there are antibodies present that bind the AAV at all. And this identifies all antibodies, and we found to be very reproducible and sensitive, particularly at the lowest levels, which are used to detect inclusion in a study or access. We've launched a program to understand how to change the prevalence of this by reducing, for example, here is the threshold for inclusion in a study in this hash line. This individual in the green is eligible from the beginning and may get treated and have no consequence. The subject B may be high at the beginning and need to have the levels lowered down to this point. And there are some interventions we're studying, particularly in Duchenne, to see what's required to do that. And certainly if there were a very high level, like in this red line, this may be more difficult to bring all the way down to the level of acceptable titers. And we found that about half of school-age children are in this category where they're either somewhat over this line, may be amenable to a therapy that would reduce their antibody titers. And certainly those that were in a study previously have very high antibody titers and are probably not amenable to conventional strategies that we're trying for this group. And we are learning more all the time about which thresholds are appropriate and how to use conventional drug therapies to make more patients, to make sure more patients are eligible. So the last question is receiving, next to last, receiving gene therapy for cardiomyopathy durable for the lifespan? So I mentioned this at the beginning, cardiomyocytes don't divide. Once the gene is inserted into the nucleus and it continues, it appears, and we've studied now, particularly primates and even some human studies going beyond 5 years. And we know, at least to that level of durability, that we expect the expression in the heart to be effectively permanent. So yes, very likely this will be permanent in the heart. If the skeletal muscle is an important part of the condition, and this is one of the challenging things for our Barth syndrome project, and certainly for Duchenne, the earlier one is treated, the less durable it's likely to be, because of the way muscle cells divide and muscles grow. A fixed amount of the product will wind up in the muscle, and then the cells that help to build more muscle will not carry the gene, and therefore reduce the effectiveness over time. We do think this is a manageable problem. It's not currently been studied in other than one study we have here at the University of Florida, where the product will be administered several times through a management of the initial immune response to allow it to be administered a second or a third time. And I mentioned the importance of this in Barth syndrome because many of the patients find that, aside from the limitations faced by having cardiomyopathy, that they have substantial fatigue. And that is the primary driver of the diminished quality of life that those patients face. And so having enough expression in the skeletal muscle will be critically important for them. And this explains why when muscle cells divide, we get extra cells that don't have the gene contained with them. We studied this in one animal model, and you can see if we measure the amount of an enzyme activity over the dose in 1 month of age, by 12 months of age, these animals had lost a substantial amount of the activity. So we know this is a problem that can be addressed, and now we hope to implement it certainly in the Barth program and ultimately someday in Duchenne study. So these are important issues because the primary antibodies preclude repeat dosing, and it does affect the impact the efficacy. So we think we really need to solve this problem. We use a combination of drug therapies that are much like sometimes used in transplant, and we think this has been the effective way to treat this problem. Here's an example of 2 patients who received this drug therapy, and actually the first study in which AAV was administered twice. So this approach has been tested in one condition, and we think this will be necessary for the other conditions like Duchenne and Barth syndrome, where we're focusing mostly on the cardiomyopathy, but for which there's also an important skeletal muscle component. So just to summarize that part, so the antibody formation against the capsid is important both to enable subjects to participate or for their secondary responses if they may need to repeat exposure later. And this pre-immunity issue is very relevant in school age and older children to enable either study participation or access to commercial product. This enables early treatment, which is now we're identifying increasingly through advanced genetic testing the cause of various forms of cardiomyopathy. And then this also allows us to consider incremental dosing, which we're not certain that every patient will respond to the same exposure, and it may be possible to optimize a given individual's response by incrementally dosing like we do with other drugs to reach the most desirable effect. And the last question is, can these antibodies be reduced to allow for entry into a gene therapy study? That's a new study that we're just undertaking in boys with Duchenne using another recently approved drug that affects antibody levels. And we do think this will in fact be very successful in including the inclusion into studies. So just to leave a little bit of time for questions, here are the take-home messages that we think this strategy to modify or add genes through AAV vectors can be successful in treating pediatric cardiomyopathy and is a safe strategy that's really long-lasting. This whole field has been popular in the last 5 years and has led to a lot of cost associated with commercial supply of the product. A treatment for Duchenne will most likely be in excess of $3 million per patient. The treatment costs for SMA are $2.1 million, and they are only on average 3 kilos. So you can imagine as we get to school-age children and adults, these costs become an insurmountable barrier unless we really think disruptively about how to make these products at a lower cost. And we're working very aggressively on that to make sure that all size patient communities, small to large, can have access. The immunomodulation strategies can block AAV antibodies, which does increase the safety of the approach and hopefully will increase efficacy as well. And that early exposure may be less durable, but we think that this immune modulation strategy will allow for repeat dosing. And this pre-immunity issue, which excludes patients from treatment, we will know in 6 months whether the current strategy we're using can influence eligibility after this new drug study gets underway, supported by actually 3 patient organizations that are focused on Duchenne muscular dystrophy. And then lastly, that AAV gene therapy can be associated with risks. We know that the innate immune response, and in some individuals, the inflammation caused by the exposure to the AAV has been a substantial risk. But we think this can be mitigated through the use of these immune modulation strategies, much like we did with transplant in the early period, which has now led to, really very successful management of anti-allograft rejection that has been, well managed now in cardiac transplant. And then these risks really justified with the potential for long-term benefit. So I'll just say a lot of what we do as part of my role as the chief medical advisor for the MDA is see patients with both skeletal and cardiac myopathies within the 150 care centers across the US. Certainly there are many centers of excellence for cardiac care where you're likely to have access to that in your community. And in my background as a pediatric cardiologist, I know that really there's expert care at almost every major medical center. So we think that as we disseminate these ideas throughout the provider community, that all patients should have access to these therapies. And I always want to make sure to thank the team that does all this work, both the clinical studies that we do and the basic laboratory research that enables those clinical studies. And certainly many thanks to patients who participate in the studies and have confidence and hope that we'll bring new treatments to their specific condition. So I'll end there, thanking specifically these folks in the lab. I mentioned Solid Bio funding the Duchenne study, and certainly our group at the University of Florida and the funders at the NHLBI. So I'll stop, and I hope we have a little bit of time for questions.
Yes, thank you, Dr. Byrne. At this time, we encourage our audience to submit your questions by using the question and answer or the Q&A box. Please know that Dr. Byrne can only answer questions of a general nature and cannot provide medical advice for a particular individual. Dr. Byrne, we do have a couple of questions. First, let's start with, what age is optimal for treatment of a genetic cardiomyopathy with an AAV vector?
So I touched on this because I think this is somewhat unique circumstance of the fact that heart muscle cells are not dividing after birth. And so if there were newborn screening for a condition that affects heart function, and the issue is really isolated mostly to the heart, then in fact, those children could be treated in infancy. And we believe that that would be effectively durable for the lifespan. So cardiac only conditions, if they were identified early, then even though we have a lot of functional reserve in heart muscle cells, it's really much more feasible to do at a young age than an older age because of the availability of the product that's used to treat the underlying condition.
Okay, great. Another question here may have been addressed during the presentation, but what if your child's DCM is considered idiopathic even after generic screen?
Genetic screening, maybe, yeah. So,
oh, sorry.
Yeah, to have a specific approach using a gene therapy strategy, the idiopathic causes of dilated cardiomyopathy would not be candidates. There was an effort to address some of these issues with an approach that was developed by a company in the West Coast that felt that improving calcium handling within cardiomyocytes would lead to improved function. There was a small study called CUPID that was expressing the protein called SERCA2A in the heart. That study didn't show a dramatic benefit, which shows you the limitations of not knowing the specific cause, because then the kind of generic therapy is probably more well managed through a drug treatment, through a small molecule drug, and not a gene therapy.
Okay, thank you. Another question is, are there any gene therapies targeted towards heart muscle function? For example, reducing muscle growth, thickness in HCM patients. And do these therapies work the same way in adults? If not, are there similar studies happening for young adults?
Yeah, I think we'll almost certainly see studies in the near future for some forms of hypertrophic cardiomyopathy. There are programs in the dysfunction of TNNT2. The Pompe is another example. That's a recessive condition, but the really unique opportunity to do also editing of specific mutations is there. And as an example of that, the NIH director, Francis Collins, has studied for 30 years the form of premature aging known as Hutchinson-Gilford progeria. And that's due to a single nucleotide change in a gene for laminin. And there is a new development by a company called Beam Therapeutics that allows the editing of that single base to return it to its normal codon. And that would be expected to then ameliorate some of the symptoms of that condition. Is an example of effectively where editing was the only strategy because of the way the mutation works. So I think there'll be more opportunity to see those effectively dominant diseases treated.
Another question came in. Are you going to do separate trials from kids that have been vaccinated versus kids who haven't?
COVID vaccination, I think it's referring to. So no, there's no impact of vaccination on eligibility for studies.
Okay. Would this be available to children with Noonan syndrome?
There are considerations of how to address RASopathies with this approach. Again, any gain or loss of function could be considered as in a way to approach a gene therapy program. To my knowledge, there's not been any groups that have pursued that yet, but it's conceivable that really any of either the syndromic cardiomyopathies or where we know that there's a recessive condition could be addressed by this method.
And how can a child become a candidate?
So there is a site called clinicaltrials.gov that identifies all the specific trials that are in the US. And if a subject or study participant meets the criteria that are set out in the clinicaltrials.gov description of the study, then there's a way to contact the study managers. And many studies have sites around the country. And also one can contact your primary caretaker or cardiologist to help mediate that discussion with the site to see if a given individual is eligible.
Okay. For HCM, the disease presents itself in late adolescence. So is it too late for AAV therapy once the heart muscle has created scar tissue and the left ventricle has enlarged and the child is in their teens or older?
A very good question. Some changes, like we know, certainly in the nervous system, when there's loss of neurons, they're not amenable to being repaired. And this may be true to some degree in the heart tissue as well. The good news is, though, we have a tremendous functional reserve in heart muscle, more than any other tissue bed. Maybe you could compete with the lungs, but some loss of cardiomyocytes or scarring or fibrosis would still lead to good level of function if there weren't any further deterioration. So a specific treatment really at any stage of the disease. And we're even contemplating that this might be relevant to patients who receive a ventricular assist device at that end stage of disease and be able to arrest the deterioration and possibly avoid transplant. So I think really at any stage in the journey that patients face with heart failure, whether they're pre-symptomatic children of an affected adult or they have more severe accumulation of a deficit as teenagers, I think they'll probably be amenable to this kind of therapy.
Okay, and we've got a question here. If you're using gene silencing approach to downregulate the poison peptide, how to avoid a further possible
haploinsufficiency? Yeah, that as long as there is persistent expression of an RNA-mediated gene silencing, this will continue to effectively eliminate the messenger RNA. There's many ways this can be used through siRNAs or exon skipping, but RNAs are made continuously because the DNA is essentially permanent within the cell. So they should have a long-lasting effect. And if that is a successful strategy for a given gene, I think it'll be durable.
Okay, great. I believe that has gotten through all of the questions. If anyone has any further questions, they can be sent to info@childrenscardiomyopathy.org and we can get them answered for you.
And as if there are other questions or concepts that need to be explained to individuals, feel free to email me.
Sounds good. We do have one that just popped up, and since we do have a little bit of time, do you think testing this in HCM is on the horizon?
Yes, yeah. So one example is LAMP2B deficiency causes a form of HCM, Dana's disease, and that study is actually underway, enrolled now 9 patients. So, and there'll be other causes of HCM which will ultimately be tested. I believe once there's a little more experience with replacing loss of function conditions, because many of the later onset HCM conditions are relatively mild, so that may not require the level of expression in some of the conditions that we're studying. For example, we're very close to launching a study in Friedreich's ataxia. This, because of the mitochondrial dysfunction in Friedreich's ataxia, there is effectively HCM. Very small amounts of frataxin seem to be effective at improving mitochondrial function, and we would expect then that those forms of HCM would be normalized after the treatment. The sarcomeric proteins may be a little more challenging, but I think there's certainly the technology exists to kind of conquer those problems as well.
Okay. I believe that answers the questions for now.
Yeah, and anyone that needs to email me can write to barry.byrne@ufl.edu.
Great. We will definitely include that in the recording. Also, thank you so much for sharing your time and expertise with the group. Dr. Byrne, we appreciate it very much. It was very informative and helpful. I also want to remind everyone that September is Children's Cardiomyopathy Awareness Month, and CCF is sharing awareness activities and information about the disease on a daily basis throughout this whole month. So please visit our website and Facebook pages, Instagram for more information. We thank you all for your attendance, and we hope you have a great rest of your day.
Thanks, everyone.